All right. Ladies and gentlemen, we are back. Third video time. I hope you're excited because I am. We're finally delving into biology, which I have a lot of expertise in, so hopefully this video will be more interesting than the other two, which were kind of just passion projects for me. But kind of sticking to the theme of climate change today and talking about a real life, tangible, biochemical example of why climate change is going to be so devastating for us. And I'm really hoping that maybe if you're a climate denier, naysayer, you're maybe thinking, there's more carbon dioxide in the air. And this is an argument that you could make, and it makes sense on paper, but I'm going to dissect why it's not the case today. You could say, there's more carbon dioxide in the atmosphere. It's like thought to be so bad or whatever, but plants need carbon dioxide. They need carbon dioxide to make glucose. Shouldn't plants be like so excited that global warming is going on? And sadly, it isn't that simple, and that's what I'm going to discuss today. Because what's actually going on? So there's actually a battle between carbon dioxide and oxygen, which is leading to some pretty tragic consequences in the next coming decades. That's what I'm going to unpack today. So, when we're actually talking about carbon dioxide and oxygen, one of the reasons why having more carbon dioxide in the air isn't beneficial to plants is that while there's more carbon dioxide to turn into glucose, carbon dioxide is sadly also a greenhouse gas, so that means it's going to increase the temperature of the planet when there's more CO2. And this increase in temperature is actually what drives this interesting switch and reversal in the Rubisco enzyme, which makes plants more efficient. So that's the enzyme we're going to lock in on today, and is really the key player in how climate change might actually mess up a lot of our agriculture. And so looking at Rubisco, there's some fun facts for you. It's the most abundant enzyme in the world. It's the start of the Calvin cycle. It's the enzyme that attaches carbon dioxide to this molecule, which eventually leads to make glucose. Rubisco, very, very popular enzyme. If you take a plant and you look at all of the proteins inside of that plant, roughly 50% of all of the protein mass in a plant is going to be exclusively Rubisco. So really important enzyme. It takes roughly 100 trillion kilograms of carbon dioxide from the air every year. So this is a pretty consequential enzyme. So let's just look into what normally happens when Rubisco is functioning. So it's the start of this Calvin cycle here. And what Rubisco does, it takes carbon dioxide out of the atmosphere and it attaches it to this molecule, or I guess you can't see me pointing, but the molecule labeled RUBP stands for ribulose bisphosphate. When you have this CO2 attached to it, you get 2,3-PGA molecules, which then through some chemical conversion turn into GA3P, which then can be used to make glucose, and the other half is used to remake ribulose bisphosphate so the cycle can continue, right? And this is what we want to happen. This is what C3 plants rely on to get their energy. And I'll get to the distinction of what a C3 plant is later. And now let's look at what happens, because carbon dioxide and oxygen are very similar molecules on the molecular level. They're both linear molecules. They're both nonpolar. So if you wanted an enzyme to distinguish them, it's pretty difficult because they don't really look that different for all intents and purposes. So what can happen is instead of carbon dioxide binding to Rubisco and adding onto ribulose bisphosphate, you can have oxygen also bind, which then, let's look at it here. When you have oxygen added, it's going to make 1,3-PG, which that's exciting. You need 3-PG. That's pretty cool. But then you also make 2-PG, which is not awesome, not cool, because the 2PG, it actually can't be used to make glucose, and it isn't really useful for anything in the plant. So what it does is that it has to recycle this molecule through a bunch of steps, uses up energy, uses up 3.5 ATP and 2 NADHs. That might mean nothing to you, but essentially, plant is using its own energy to reconvert this molecule into ribulose bisphosphate, and along the way, you also lose a carbon dioxide. So you're losing carbon, you're using energy. It's not awesome. C3 plants don't like photorespiration because they're basically just getting exploited. They're using, let me see here. Oh, I have it written here somewhere. Photorespiration decreases carbon fixation by 25% by virtue of using that energy that could have gone into making glucose in what is a so-called futile pathway of photorespiration, where you're converting 2PG into the starting material again, and you're also losing a carbon dioxide and losing energy as well. So photorespiration really doesn't give a lot to plants, right? Unless you're a C2 plant, which here I'll make the distinction between C2 and C3 plants. There's also C4 plants, which I won't mention. But C3 plants are what we rely on day to day, and roughly 50, or not 50, 80, 80% of the plants we eat are C3 plants. So, for example, wheat, rice, and soybeans are all C3 plants. And what this means is that C3 plants rely on the Calvin cycle. They rely on carbon dioxide, making it into and binding to Rubisco so that you can make glucose down the line. And then you also have C2 plants, which C2 plants are what you'd recognize as plants that live in the desert, right? Like a cactus or something, right? They're used to low water conditions, high heat, and how do they survive here? So it's actually interesting because they rely on photorespiration. They know they kind of just take the loss. They're like, well, I'm in a place where it's really hot. Rubisco is favoring oxygen, and I won't get as much carbon dioxide. So how do I still make glucose? So what happens is that when you're undergoing photorespiration, right, you have the oxygen binding, you make 2PG, which then you have to convert back. But when you're converting it back, you lose a carbon dioxide. And in C2 plants, they have carbon capturing mechanisms where they take this carbon dioxide and feed it back into Rubisco so they can at least make some glucose from that oxygen, which is normally a futile cycle. But the sad thing is that most of the plants we rely on are C3 plants, so they don't have that mechanism to counteract photorespiration. And I probably mentioned this already, but increasing heat loosens the discrepancy between Rubisco wanting to bind carbon dioxide and oxygen, and prefers oxygen in hotter environments, which is why crop yields are going to suffer greatly with these increased temperatures. Let me see where I'm at here. So I'll publish all of the, or I'll put links to all of the papers that I read in the description if you'd want to check them out. But one of these papers here is talking about how right now the atmospheric carbon dioxide level is at 425 parts per million per volume. And then they're estimating that if we reach 600 parts per million of carbon dioxide, it's going to equate to roughly a 3.7 Celsius increase in temperature. And even though there's more carbon dioxide in the atmosphere, the increased heat is going to lead to around 8 to 20% loss of efficiency of all C3 crops. So imagine in a yield, some farmers growing soybeans or wheat or rice, they're going to get 8 to 20% less of what they used to just by virtue of the temperature, making it harder for plants to grab carbon dioxide and make glucose, which they need to survive. And, this seems like a definitely grim situation, but luckily there are some workarounds in terms of bioengineering to try and make C3 plants more able to survive in a hot environment. And one paper I'm going to talk about, they're actually modifying tobacco plants to try and make them more efficient to grow in hotter conditions. So what they're doing in this paper is they're inhibiting this one transporter, which is called PLGG1. And yeah, I just gotta find this figure here. So yeah, what they're doing, they're inhibiting this transporter, which you see on the picture is crossed out in green. And what it's doing is that it's preventing glycolate from entering the peroxisome or mitochondria by preventing the transporter which normally takes it into the peroxisome, in and out of the peroxisome. And this, in this green dotted arrow, this is the cycle that then takes over. You're essentially shutting the door in the face of the plant. You're saying you can't move glycolate anywhere. It has to stay in the chloroplast. So it goes through this other cycle, which is roughly more efficient than going through regular photorespiration. And they found that there's a 5 to 18% increase in the Calvin cycle or carbon fixation at 40 degrees Celsius when they block PLGG1 using RNAi, which I'll probably end up doing a video about eventually. But essentially they're blocking this transporter, they're blocking the futile cycle of photorespiration. Instead, the plant uses some other mechanism, that green dotted arrow on the diagram that I showed, which increases the yield of the plants because that's less of a futile cycle. They can get more reward out of having this cycle by virtue of blocking photorespiration from happening. So this is one way. This isn't the only way. There's a bunch of ways you can modify metabolic pathways to try and get a different outcome. This is just one paper that I'm showing to you. But the reason that we actually have to modify this pathway is that it's actually kind of inefficient in terms of enzymes. So what I mean by this is that Rubisco, compared to other enzymes that we know exist today, is actually pretty inefficient in terms of how much, it's pretty inefficient in terms of how much carbon dioxide it can work with per second. So Rubisco can take up around 1 to 22 molecules of carbon dioxide per second, which doesn't sound that bad until you consider alcohol dehydrogenase, which works to get alcohol out of your body and metabolizes it, which works roughly 1,000 times per second. So you can see this enzyme isn't working as fast as alcohol dehydrogenase. And one of the reasons behind this, and also the reason why there isn't as much specificity for carbon dioxide, is just a virtue of how this enzyme evolved. And what I mean by this is that when life first came about, roughly 4 billion years ago, stuff started living and whatever. I'll definitely be talking about abiogenesis in upcoming videos. But you have these primitive cells, then they start photosynthesizing, right? And these first photosynthetic cells are having a great time. They can capture energy from the environment. They get a molecule which they can then use later on for energy. But what isn't great is that now when these photosynthetic cells start popping up, they're adding oxygen into the atmosphere. And the problem with that is that on early Earth, there was no oxygen in the atmosphere. So it was just carbon dioxide, methane, maybe some other gases, but there was no oxygen. And so there's what was called the Great Oxidation Event, which is really tragic. I mean, I guess it was all bacteria dying, so I don't know how tragic it was, but a lot of stuff died in the Great Oxidation Event just because oxygen is a highly reactive molecule, and even today we need oxygen to breathe, but oxygen is trying to kill us every day. And what I mean by that is oxygen, it can form superoxide radicals, which is basically a really reactive form of oxygen, which can then attach to fatty acids, DNA, proteins, and inhibit normal function of cellular processes. And don't really worry about this because our bodies already have lots of mechanisms to counteract this, but it's just an interesting fact that oxygen is actually one of the deadliest molecules and led to a mass extinction when there were these photosynthetic organisms that started creating oxygen. So there was this race for some organism to be able to counteract that toxic effect that oxygen has. And since this photosynthesis was evolving for such a long time without any oxygen in the atmosphere, there wasn't really an evolutionary push to prefer carbon dioxide, because then oxygen came into the atmosphere and it was like you already have this enzyme that's good at taking up carbon dioxide enough, but now you introduce another molecule that looks exactly like it. You need to wait for longer time, like after that, for there to be more specificity. And so we can kind of work on this ourselves and I guess try and increase the specificity of Rubisco. And there's some work that's being done right now to actually try and see how we can modify this enzyme so that it becomes more selective towards carbon dioxide. And what's been found so far is that if there's the active site of Rubisco, if there's a net negative charge, it doesn't prefer CO2 as much. But if it's a net neutral, no charge, then carbon dioxide is more likely to bind than oxygen. But one of the problems with this is that it's pretty hard to modify Rubisco just because it's made partly in the nucleus and partly in the chloroplast. So it's hard to genetically modify, but it's another route that we can go about trying to get C3 plants to be able to adapt to the unnatural environment that we're living in right now through climate change. And that's really the end of the video. I really appreciate anybody who stuck out this far. I was just trying to, I guess, sprinkle a little bit of hope into the unprecedented, scary time we're in right now. But I will say there's a lot of very smart people working on this problem, and I have a lot of hope for the future that we'll be able to pull through. And I think with all the ideas that we have pumping right now, I'm sure that we'll be able to counteract global warming in some way. And then, if by any chance you're a climate denier and you're still watching this, I just have one question for you. And it's, would you rather us address climate change or necessitate that we have bioengineered plants because naturally occurring plants won't survive the environment that we're going to have in the next couple decades? Because you can't have it both ways. You can't deny climate change and then also say bioengineering is bad. That's like we dug a hole for ourselves, and now you're taking the ladder away from us. You can't. I don't know what to tell you, bro. If you don't agree with climate change and you also don't like bioengineering, you are the worst type of person. And I'm sorry, but something needs to be done. And I'm really excited for what research is going to come out, because I think we are going to come out of this stronger, but it's going to take a lot of effort and a lot of research to really go into how climate change can influence various factors of our lives we never think about, such as the yield of plants which we rely on to eat every day. Hope you enjoyed this video, and I'm excited to say that I have a lot of videos planned for the future. Like I said, I do have a lot of obligations outside of making videos, so I don't know how frequently they'll be coming out, but I do have a pretty decent stack that I'd like to get through. So if you are patient, in the next coming months I'll definitely have more videos coming out. Have a good one.